The first nucleotide that we're going to talk about is guanosine. And just real quick, we're talking about nucleotides, but there's not really a word for nucleotide specifically. It's something consisting of a nucleoside and a phosphate, or one or more phosphates. So what we're going to talk about is guanosine, monophosphate, diphosphate, triphosphate, and each one of those is together. The guanosine plus the phosphate is the nucleotide. So what they're showing here is the nucleoside without any phosphates. And the base, the nitrogenous base that's added on, is just the guanine. So when we're talking about DNA or RNA and we're listing out letters, the G is referring to guanine, and that's referring to just the base on the larger nucleoside, which is on the larger nucleotide. And just real quick, when it's missing an oxygen here, it's a deoxy. So like this is a deoxyadenosine monophosphate, and if it was guanosine, it'd be deoxyguanosine monophosphate. So I already searched for it here on MolView. You can see the ribose 5-carbon sugar here with this carbon. The nitrogenous base over here, which is guanine. You have the carboxyl group here, amine group here, and these are the three positions that bond to other nucleotides in DNA and RNA. So this is the 1' carbon, 2' carbon, 3' carbon, where you'd build the chain of nucleotides. 4' carbon, 5' carbon that has one or more phosphates, or if it was in a polymer, this would be connecting to the 3' of another nucleotide. And then if we wanted to make this in DNA, we would just have to remove this oxygen here and be deoxy at the 2' carbon. But let me just put that back so it matches this 3D representation over here. You can see the phosphate here, the ribose. This is part of the sugar phosphate backbone of DNA or RNA. Again, this is specifically RNA with the oxygen here. And then you have your guanine base here, which is a purine with the double ring formation. And then the 3 positions that it can do hydrogen bonds with to another nucleotide. Cytosine base. So before I was in my amino acid playlist, I was drawing the amino acids, but I think with the nucleotides, it's probably easier just to bring up what they have and remove part of it. So kind of deconstructing this, I'll kind of chip away at it from the phosphate side. So you have your phosphate attached, and these would actually be negatively charged oxygens missing hydrogens in a cellular pH. And then if this was in a polymer, there'd be a ribose attached to one of these with it, leaving a single negative charge on the molecule here. But let me just kind of start chipping away here. So removing your phosphate here, you're left with just guanosine. And here's your 5' carbon, 4', and that's where the oxygen connects the 1' to the 4' carbon. And then here's your 3' carbon, and then here's your oxygen that would be present in a RNA and missing in a DNA molecule, and your 1' carbon. I'll leave that, I guess, kind of just attached like this. So I think, let me just double check the numbering. So here's a purine. The base is going to connect to the sugar, the ribose, at this 9 position. So here's the 9 position here, which means this will be 3. So here's 3, so this means it's numbering here 1, 2, 3, 4, 5, 6, 7, 8, 9. And then this is 1'. So they number the base and then they number the ribose using primes to distinguish them. So at the 2 carbon you have this carboxyl group, and at the 6, I think, 6 carbon here you have a amine group. So that's kind of just stated on this page here. C6 carbonyl group and C2 amine group at C2, and then N1 is going to also form and this hydrogen bond to another nucleotide. So guanosine triphosphate is used in metabolism. It's energy storage, and it's part of energy transfer in the cell, regardless of DNA and RNA and data storage, genetic information. So during translation, when the ribosome is translating messenger RNA into polypeptide, it uses GTP during this translocation part of it, where the transfer RNA gets shifted over. It requires a conversion of GTP into, I think maybe GMP, guanosine monophosphate. It might be just the diphosphate, though, depending on how many phosphates get removed in that reaction. Same with these G protein coupled receptors. It's a signaling molecule to get information through a cell membrane. So when a molecule attaches to one side, on the other side of the membrane, a guanosine triphosphate gets converted to a diphosphate. Other way around, there's a guanosine diphosphate, and it gets converted to a triphosphate to kind of activate the G protein inside the cell. And then there's also the cyclic guanosine monophosphate, where this phosphate binds back to the ribose sugar on the here's the 5' carbon, so that was already bound to the phosphate, and then the phosphate also binds to the 3' carbon here. So it kind of just creates this cyclic loop here and prevents the nucleotide from forming a chain because the phosphate's used up and the 3' hydroxyl group is used up. So that's a signaling molecule. It's a second messenger, meaning it's some hormone or something was the first messenger, and this is later in the chain of messages being converted from one form to another. So I already showed the purine here, and there's a few functional groups that I mentioned. Carbonyl group with the double bond to the oxygen, and amine with the nitrogen floating off in space. This is probably another kind of bond or functional group here that forms in the this center position on the base pairs. So here you can see guanine and cytosine forming hydrogen bonds with each other, 3 bonds. And we'll talk about that later. The last thing I was going to mention is just that that it was named after guano, bird droppings. I didn't mean to click on that, but it was discovered, or even named in 1846, and discovered from seabird excreta. All right. On that note, I will stop the video.